Question you could ask right now. Anything? Okay. Yes.
Yeah. I have a questions about cycling. When I revise my lectures at home, I see that some kind of graph of cycling. It had just one member like cycling A or E. Yeah. What other graph of cycling like cycling D? It had three isotypes like cycling D 1, 2, 3. Yeah. So I wonder that if three on kind of these cycling these present in the cell, or it is specific for a type of cells. Yeah. Okay.
Yeah.
And if it is specific for each type of cell, whether it's the target of a specific growth factor to stimulate the cell cycle, and if it is true, whether people had developed some kind of drug to specific inhibit some kind of cycling to limit the side effect.
Good. Okay. That's a very good question. I think he was asking whether other subtype of cycling, for example, cycling D1 or D2 or D3, may be similar to what we just said, or those cycling process. That's a very good question. Obviously, you have read some of the other type of cycling that could be, they do exist, and then the profile, we give it to you, not complete. So basically, I think if those are subtype cycling, they more or less behave the same, okay? But in certain cases, maybe have some specificity that a little bit different, but not a whole lot. We know that a lot of protein and a lot of isoform, which I'm going to demonstrate to you some of the isoform we have, for example, 1433. They actually behave a little bit different, even though the sequences are very similar. And the cycling D, in this case, majority they behave the same. But you asked a very good question. And you certainly could search for literature to give you a good answer to that, okay? And very good, keep on asking that kind of question. And that's really a good spirit for doing research. You wonder, if anybody have done something like this, or if nobody have done that, you could do the same thing. So you're asking second D1, second D2, or D3, or maybe B1 or B2 or B3, or whatever. Do they all behave the same? Maybe, I will assume they behave the same, but maybe I just didn't know that someone actually had not done it, okay? So you could do the experiment or research and to demonstrate the differences there. So that's an important spirit for all of us, and we want to be a good scientist, okay? Good. Any other question? Okay, if not, let's get started to continue. And I will speak very slowly, okay? And so don't hesitate to ask me questions, interrupt me, okay? And I know because of some of the experimental detail may be a little difficult to some of the people here. And it's important I want to introduce this to you because if you just read the textbook, for example, the textbook we give it to you, it's just very descriptive to summarize some of the results. But in the end, you still need to go back to the original literature to find out what exactly they say. And then in order to understand what exactly they say, you need to know that it's a little detail, okay? For example, how they do IP Western, how do they do the assay, by doing FACS analysis, how we talk about using shRNA, we talk about using siRNA to do the research. All these experimental details is very important for you to understand further. So even though you encounter some difficulty in understanding some of the experimental detail, you know, help yourself, ask your colleague, ask your friends, ask your classmates to see someone know better to explain to you. You know, you should make effort to understand those, and that will extremely help you to appreciate what has been done in the past, and then what we could look forward to in the future. Okay. So we stopped here yesterday, right? So I'm adding up a little bit some of the other things. Here, as we just mentioned about 14th Street, okay? This is one of the areas my lab has spent quite some time and effort in this area. It's certainly a very interesting protein. But let me just give you the history about this protein. It has a very strange name, 14-3-3, okay? You know, it's kind of weird why the protein been named that. But some of them other protein also giving the name according to certain characteristics they have. For example, we talk about P21, P27, P57, P53. Okay, this is a very simple nomenclature. And the reason that was giving these names is just based on their molecular weight. Okay, these protein has a 57 kilodalton or 53 kilodalton. Very simple. Okay, and sometimes the protein was given a name with an important developmental feature. Okay, when they involve in developmental process, or they involve in the certain biological function, and we talk about CDK, CDC, that was based on the yeast genetic study. We know that they involve in cell division cycle, so that's the reason they are giving the name CDC. Okay, so that's very. I just want to give you that kind of all the gene has some history behind it. So it's very important when you study certain protein, try to understand the history behind it. Okay, that will make you appreciate more why originally the protein or the gene was identified. So 14-3-3, very interesting protein. Okay, it was originally identified in the brain extract. Okay, so the scientists, the biochemists, try to understand what's in the inside of the brain extract. What kind of protein are present there? Okay, are present in the brain extract. So they run a 2D gel, okay, try to identify the protein. Okay, we run a lot of 2D gel, and some of you probably know what is 2D gel about, based on the molecular weight, based on the alloy extraction focusing point. Okay? So different spot, different protein will based on these characteristics, they will move to certain area of the 2D gel. Okay. And then the 14-3-3 sigma or 14-3-3 protein was identified because the scientists try to using 2D gel to identify different proteins. And so they find out in the 2D gel number 14, and they have the coordinate of 3-3. Okay? Some of you know coordinate, right? If you've done based on the mathematics, you should know the coordinate. So the coordinate of 3-3, and it is run on the protein was identified in the number 14 gel, and then it was a coordinate of 3-3. So they named it the 14-3-3. Okay. And then right now, if you look at the literature, so many literature describing the 14-3-3 biological activity involving many different kinds, including signal transduction, including developmental process, including the neuronal development. Okay? So right now, this protein right now has a different kind of function. Here, I just want to introduce to you the 1433-3 protein actually has a lot of different isoforms, as you can see here. At least 7 isoforms was identified. Okay? But you look at the sequence, they are very homologous. Okay? Almost you could not identify because you're only about within 2 to 5% difference. These isoforms are very close to each other. They are very homologous. Okay? But in detail, when we do more study, we found that there's some difference behind this. The sigma form is the one very unique, and then we've been studying for the past 10 years. It's very interesting. This 1433 protein, but then later on, identified as a phosphorserine or phosphorthreonine binding protein. Okay, meaning that this protein will bind to the target that has been phosphorylated on serine residue or threonine residue. Okay, so the binding motif has been identified here with this motif, and then including you should learn how to pronounce this Greek alphabet. Okay, anybody know how to pronounce this one? Raise your hand. What's that? This one. This one is delta. Is this one delta? Anybody want to say yes or no? Yes. Yes, right? So, and how about this one?